My name is Marissa Jones. I'm the Business Development Manager for biocrates Life Sciences. I have my Ph.D. in analytical chemistry, so we can talk science or we can talk business. But what you guys might not know is I am also a mom, and I'm going to start this talk off a little bit counterculturally, and I'm going to tell you a story about my son. So in short, my son is 15 months old, and he has a favorite toy, and you'll never guess, it's a hammer. And he uses that hammer as an alarm clock, he uses that hammer as a button pusher, or even as just a way to chase around the dog, you know? And I think, like my son, we all have a favorite tool. And not that I'm comparing us to a 15-month-old for sure, but I think we all have a reason to like our favorite tool, whether it be genomics, whether it be proteomics, whether it be some type of modeling or anything like that. We all have something that's tried and true and that we know works for us. And today I just want to open up your mind to adding a new tool to your toolbox. And I'll start out with talking about why metabolomics and looking at crossing the translation barriers, and then looking into what biocrates has to offer and how we help with that, and then a quick case study and summary. So I'm sure I don't have to tell you guys about omics, but I think that, we've all we don't have direct experience with a single omics. We know it in theory, but we don't necessarily know where it fits with what we know. And so I wanted to talk a little bit about why metabolomics matters. It's one of the many omics, but what's unique about it compared to genomics or transcriptomics or even proteomics? So if we look here, we can see that in the omics cascade, you've got your genes, and then it moves on to your transcriptome, and then onto your proteome. And you can see that many things affect your metabolome. But what you can also see is that it tells you what could happen or what could make it happen on the side of the genome, the transcriptome, the proteome. So it tells you who's there, but not necessarily what they're doing or what the outcome of what they're doing is. And that's where metabolomics comes in. It tells you what actually happens and gives you the final pieces that are the closest to the phenotype. And this is influenced by many other areas, including the microbiome. I'm sure you guys have all heard of like the gut-brain axis or the gut-heart axis or all of these different axes. And what the metabolome allows for is a functional readout of all of these different inputs. And just to give a little background, you guys know the issues that you're facing in pharma, but I think it's important to put into context how metabolomics could make a difference here. We know that 1 in 1,000 compounds reach a clinical stage, right? And that what it takes to get there is billions of dollars, and what it takes to get there is a lot of time. And we also know that only 1 in 5 of these compounds succeed in a clinical stage. And even if it succeeds, it may not be useful for all patients. In fact, you have a non-response rate of about 1 in 3. And so what can metabolomics do to help improve this? If we look at the clinical stage, if we look at when we're first looking for a compound, what metabolomics can do is it can identify drug actions beyond the immediate target. So oftentimes when I say targeted metabolomics, I think that people are kind of picturing a telescope and they're looking at like a single metabolite or a pathway. But we at biocrates, when we say targeted metabolomics, we don't mean only look at this metabolite. We are looking at a broader view. What we mean is that we're quantitating and identifying, and we're looking for over 1,000 metabolites. So it's giving you that kind of broad, untargeted flavor with the targeted backup. And this allows for improved prioritizations of promising compounds. Now, if we look at that translation from the compound to the clinical stage, there's a lot of steps that go in there. But the beauty of metabolomics is that it's highly conserved across species. So let's say that you're going from mouse to man. You don't need a very large sample volume. You can get, with only 10 microliters, quite a lot from metabolomics. Or most of our kits, you only need 10 to 20 microliters of plasma or biosample. And what you're going to see in mouse, obviously there's going to be some caveats, like some bile acids that you only see in mice versus in humans, etc. But largely, it's highly conserved. Secondarily, you're getting a more accurate replication of pathophysiology because you're that much closer to the physical change. So if we take a look at the third figure on the right there, we have a lack of a suitable response for biomarkers. And oftentimes when I talk about metabolomics to pharma, they're thinking that I'm going to tell them that you're going to turn a metabolite into a drug and that is going to solve all your problems. And I'm here to say that that may not be the case that you're going. What I'm trying to propose is that you look at metabolomics as a way to see what's changing and as a way to stratify your patients. Because I don't think it's always that the drug isn't working, it's that we're not targeting the right population for it to work most effectively. So you can have comprehensive patient phenotyping, as well as integration of hereditary factors, as well as lifestyle and age. Just as we showed in the last slide, that metabolomics is impacted by many different areas, that this is important and that you can see that functional readout of those changes here. So if we look at the pharmaceutical R&D workflow, you guys are a lot more familiar with this than me, but in short, I think it has 4 main steps. The first being that you've got discovery, then you've got preclinical, then clinical, and approval or market. And I think that metabolomics could make a really big difference here if we take a look at cell lines, because you can have more parameters for prioritization, you have replication of academic research, and you even have a functional understanding of drug action. Now, this sounds like a great promise, right? That metabolomics could really make a big difference. But if anybody's ever worked with metabolomics data, we know that it's complicated. We know that it's hard to know exactly what's coming out. You're looking at all these log2 fold changes. You're looking at this putative identity. What biocrates tries to do is it tries to make sure you never have to say log2 fold change again. And it makes sure that you don't have to say that, well, I don't really know what this metabolite is, but according to accurate mass, it's this. We try and avoid that. And we also try and put your metabolomics in context of what other research you're seeing. So we have this cool thing called Metabo Indicator that I'll talk about a little bit later. But in short, it's biologically relevant sums and ratios tied to literature. So then you can get real actionable insights out of your metabolomics without having to hire a bioinformatician or to learn R yourself. You can get a lot of information out of what you're looking at. So if we look at the second stage, if we look at animal models, we have more sensitive readouts to molecular toxicology, histology, assessment of organ function and recovery. There's many areas where metabolomics could make a difference. And it's representative for human disease because once again, metabolomics is highly conserved across species and you only need that small volume. So if you're doing a study today and you're looking at a preclinical study that you're not sure if it's going to go on to a clinical study, but you want to be able to compare your data across time and across space, that's what we do really well. So sequential sampling, it reduces and refines the animal experiments that way. So the third step is clinical development. And if we're looking at patients, one nice thing about what metabolomics can do is that it helps you identify likely responders to a drug. So your drug doesn't fail because of it's targeting the wrong market. I think that's huge. And if it doesn't fail fast or fail slow, like that's a big difference in terms of money. So I think that with metabolomics, hopefully you can fail faster or not fail at all. And understand the standalone approach of adding other types of information. So the beauty of metabolomics is that it integrates really well with other omics. So let's say that you want to do a proteomics and metabolomics experiment. Now, I think there's a lot of advancements happening on the data analysis side for sure, but we have some solutions for that as well. And just giving you actionable insights into patient care. And the last area, the part that the scientists don't always often deal with when it comes to the approval or the market and that kind of thing, we try and make it so that you have that quantitative test that you can use on both sides of translation and that you have a globally available technology. So you're not sending off your samples and having them run somewhere else and getting back a 2-page report and then expected to do something with that. You have your raw data, you can run it here at AbbVie and you can compare it to maybe Europe's AbbVie location because the quantitative kits themselves, you can integrate all of that data. So if we take a look at translational research, I know that unless I give an example, it's all just talk, right? So if we look at it at this example where they're trying to converge across preclinical and clinical developments. I'll go into the details a little bit later, but I wanted to kind of highlight what metabolomics can do for you on each side before I go into that example for you. So it allows for improved translation on the preclinical side, like we just showed in the last slide, and that highly conserved low sample volume. You get added value of new targets regarding academic research that you can replicate, because we all know that in the pharma world that when something works, it becomes a trade secret, which is fantastic. But at the same time, being able to take some of those academic articles and try and just get one step ahead is really important. And identifying metabolomics vulnerabilities helps you to learn whether your drug is going to fail. And if it's going to fail, have it fail faster so you save the company money, because a failed drug versus a successful drug, it's still billions of dollars, right? So looking at the clinical development side, maybe you would want to look into stratifying your patients, looking at those preclinical metabolomics biomarkers, or maybe you have a companion diagnostic based on a biochemical response to target a specific expression that you might not be able to see with another omics. So that just kind of gives you an idea of what kind of translation that could look like. And with this technology, you can have translation this way as well as back to the preclinical stage. So you have a full feedback loop rather than just a single direction. So if we look here, this isn't the example that I was alluding to earlier, where they're looking at mouse xenografts and trying to put these technologies into a phase 1 clinical trial. So this is looking at ICR London. And they have multiple papers where they're working on this, but they were looking at drug candidates and looking at interacting pathways that they couldn't distinguish based on what they see in genes. They were compound-specific, dose-dependent metabolic responses, and they each had different biological effects. So they needed to be able to identify what was happening, not just who was there. So for the proof of concept here, they've got a translatable therapy monitoring dose escalation studies, and they're able to go from mouse to man with these. So they've did a mouse xenograft model, and then now they're headed towards this phase 1 clinical trial in humans. And they're using metabolomics as that translational pharmacodynamic readout of the biomarkers that they're seeing. And if you look on the bottom here, what you're seeing is a lot of colors, right? But you're seeing the drug targets. So the drugs are targeting MEK, PI3K, and AGC kinases. And then depending on what the therapeutic response is, there's different affected metabolites and signaling molecules as well. But the affected metabolites are allowing them to see which biological effect that they're going to see. So whether it be multiple effects regarding vasodilation going through nitric oxide, or whether it be tumor cell growth or proliferation, looking at it through the lipids pathway. And they're able to identify these pathophysical readouts based on the metabolites that they're seeing. And if you want some more information on this, these are quite a few citations where they're using this general workflow with the xenograft, identifying the metabolites of interest, using them as a functional readout to identify before they see a physical outcome. And so here's just a few examples of that, and I'd be happy to send them to you after the talk. So next, we've talked about my son's favorite tool and trying to open up your toolbox. We've talked about the translational barrier, the challenges in pharma, and now let's talk a little bit about what biocrates does, right? Now that we've established what the challenges are. So in short, I think that it's important to recognize a bit of our heritage as a company. Oftentimes you'll see biocrates and you'll see something that looks like biocrates. And that would make sense, right? Because the little boxes that come to you and you run metabolomics with them. But in short, our name actually comes from the merging of biology and Socrates. So we try and get at the philosophy behind the biology. And we're a market leader in fully integrated mass spectrometry LC-MS-based kits. So we're using mass spectrometry and LC to be able to look at these metabolites. And we have a kit technology that's relevant across many different application areas. It's not just for oncology. It's not just for microbiology. Like I said, metabolomics is highly conserved, and you can see many of these different applications through that. And what we do is we develop produce and sell these kits for quantitative analysis of metabolites. So what you get out is a list of the metabolites and their concentrations, as well as what you can do with them with those metabo indicators. And we have a global community. So as I said before, regardless of where your kit is run, you can integrate your data across. And regardless of where it's run, if you have data analysis questions or you want someone to analyze your data, we have experts in-house for that beyond what you may have here. Or if you want to scale up and you're doing a large study and you might not have the mass spec bandwidth to do it, we also have a global network of certified labs that they get the same results that we would get at our headquarters. And just to give you an idea, we have a headquarters in Innsbruck, Austria. As you know, it might be fantastic for skiing there, but we also do great science. And we have a US team as well, which I'm a part of. So I think oftentimes people are thinking about, okay, this sounds like a great technology, but I need the short version, right? So the short version is there's three main reasons that people tend to work with us. It's standardized and quality controlled. So you're having harmonized data, you have great data storage, you're not worried about, is the result I'm seeing real? You're only worried about on the biology side, what you're doing to control there. But on the analytical side, you've got CVs of less than 10%. You've got quantitative and reproducible data. So comparing across instruments, across labs, we put a lot of work into this. We have many ring trials that I'll show you a couple examples of later. But you can pull that data. And that data is valid regardless of if it was run 4 years ago versus if it's run today. You can compare it across. You also have local available platforms, like in Chen's group. If you wanted to run metabolomics here, you don't have to send samples out. There's a place to do it in-house. And with the pharma cuts going around, that's always nice to be able to have a cost analysis where it's done in-house. And the biggest thing I think that most people see is that you have total ownership of your data. We don't own your data. We don't keep your raw files. It's yours. And if you want your raw data, you get your raw data. It's not a matter of, here's a 2-page report, hope you can figure something out from it. And we have many different options for what you can take a look at. We mainly talk about our kits, but we also have a contract research business as well, with many specialized assays of what you would more think about when you're thinking about targeted metabolomics. So like a tryptophan assay or a steroid assay or an acylcarnitines assay. So we have large profiling kits. As I said, it's much larger. There's 1,019 metabolites with over 470 of those metabolite sums and ratios. And then you have more specialized kits. So if you're wanting to run high resolution, let's say that your group only has a high resolution mass spec and you don't want to work with Chen's group, you could also run a kit on that as well. Or let's say that you only want to look at a small subset of metabolites. You want to look at only like 180 of them. We have a kit for that as well. And let's say that you later on think about, okay, I have identified these specific metabolites. How can I make this into a companion diagnostic? We do have experience there as well. And the nice thing about biocrates is they hire PhDs like myself in our sales roles. And if you ever have a question about project design or bioinformatics or data analysis, I can certainly put you in contact with one of our experts, but you can speak to someone like me right away. And then you've got support for global trials locally at your own location or with a CRO partner of your choice. So if we take a look at the different products that we have, there's mainly 3 levels of the kits that I think that are most likely used in most of pharma applications, and that's these 3. And it's kind of generational in that the oldest one is the P180 kit, and all the metabolites in that one are in the Quant 500 kit, and all the metabolites in that one are in the Quant 500 XL. So it just depends on what you want to target, and it depends on what you're after biologically. And, you know, for example, if you're looking at maybe only a few small molecules and lipids, you'd go with your P180. If you're looking at quantitative metabolic profiling or maybe a host microbiome interaction, maybe you're looking at urine, you wouldn't want a lot of lipids for that. So the Quant 500 would be the way to go. Now, if you wanted to just see as much as you can see, look at functional understanding, try and get as close as you can to untargeted metabolomics level, then you'd want your Quant 500 XL kit. So that just kind of gives you an idea of what they offer. And this is just an example of many of the different metabolites classes that you can see in one of our kits, we try and keep it pretty broad so that oftentimes people are going on some sort of fishing expedition where they say, I want to find out the difference between group A and group B. I don't necessarily know which pathway is most important to me, but I know that there's something with metabolism that's different because I'm not finding it in the genes. I'm not finding it in the proteins. What's different? What's happening? And so that's when people typically go here. And there's many different applications, but that just gives you an idea of what you could see. If you're used to seeing a pathway, here's what our coverage looks like. Happy to send you just a quick file of this later. But in short, we've got broad coverage as well as quantitative data, which I think is really important for the type of work that you guys do. So last but not least, I wanted to give you an example of a case study. So here we have a modulation of therapeutic response. And this is based on a paper in Nature. And in short, they're looking at a polychemotherapy drug treatment in antibiotic-free patients with pancreatic cancer, which the outcome for pancreatic cancer is pretty poor. And so when you have to make a decision, you have to get it right, and you have to get it right fast. But what they're seeing is what we typically see, which is that there's a large portion of non-responders. And in this case, it's actually over 50%. And so first they're thinking, okay, so I'm seeing a non-responder, I'm seeing a responder. Let's just take a look and see if it's on the gene level. And what they found was that they couldn't tell a difference between their responders and their non-responders on a gene level. And so then they said, okay, well, we know that antibiotics make a big difference in chemotherapy. So could it be that it's on the microbiome level, right? Could we change something in the gut? And what they found is that they saw a difference in the bacteria that were in the gut. They were seeing, okay, 3-IAAs producing bacteria were higher in responders and lower in non-responders. So that gives them an idea that the 3-IAA was important. And what they did then is they said, okay, let's take a look at this in mice. Let's do an FMT transfer, and let's see what we can find in terms of the diet. So here we have on the right-hand side, their rough experiment where they've got a human where they take an FMT and put it into a mouse, and they're doing the polychemotherapy. And then they've got long-term treatment with tryptophan diet because 3-IAA is a downstream metabolite of tryptophan. So they're thinking, oh, maybe we can just increase tryptophan in the diet because that's pretty easy to do. Maybe it's long-term, maybe it's short-term. And what they found is that in people that were already responding to the chemotherapy, if they had a short-term tryptophan diet, it increased their efficacy. But long-term, it didn't make a difference. So next they're thinking, okay, what about those non-responders? Is there something that we can do with the tryptophan diet? Is there something that we could do with making the gut more hospitable to these bacteria? Or is it really just the 3-IAA? And what they found was that long-term tryptophan increasing in the diet didn't work. Short-term didn't work. Making the gut more friendly to the bacteria didn't work. The only thing that ended up working for them was this 3-IAA supplementation. And that caused an increase in efficacy for their chemotherapy. And so, in short, to be confirmed in a clinical trial, because once it becomes a clinical trial, it becomes trade secret. But in short, what they're finding is that it does make a difference. And this is the kind of thing that I'm thinking about when it comes to metabolomics and pharma, is how can we make the drug targets that you guys have more effective to a wider population? Could there be a supplementation factor? Could there be a pre-test? I guess you guys probably have a more technical term than pre-test, but a test that you give to someone to stratify your patient population. So as we see, this companion diagnostic could allow you to predict response. It could predict non-response toxicity, and it could allow you to change your therapy in those decisions where it really matters, right? So those quick decisions about whether someone lives or dies based on the type of drug that they get and whether it works for them. So specific drugs for patients, which drug fits the phenotype test, and then maybe a co-medication or a combination of these drugs would be best. And repeated analysis for treatment monitoring. So in summary, there's a couple of things that I wanted to leave you with, but I think the biggest thing is let's open up your toolbox to include some more metabolomics. Metabolomics could be your next groundbreaking tool. Just like my son enjoying his hammer, you could enjoy metabolomics just as much, and it could apply in many different areas for you. And it could help you overcome that R&D translational barrier, maybe understand drug mechanisms, improve patient stratification, identify biomarkers for success, or reduce late-stage attrition. And biocrates wants to help you do that and wants to help provide the tools that'll give you that targeted, quantitative, and highly standardized results that you need to overcome this barrier.